Thanks for inviting me to come here. It's a little bit intimidating because I sat in those seats for many years, but I haven't been here for a little bit more than 40 years, so it's a pleasure to be back at University of Washington Medical Grand Rounds. And I'm especially honored and humbled to be chosen as the John Butler Lecturer. I knew John, or JB, reasonably well. He was the division chief in Pulmonary when I was here as a resident, but I actually had my closest encounters with him back at UCSF when he came to give talks in our science conferences, and was nice enough to spend an hour with several senior fellows and junior faculty members. So I do have a few disclosures. I'm a co-founder of a company that's developing anti-fibrotics and inventor on several UCSF patents relevant to what I'm going to talk about. But fortunately for this purpose, none of them have seen the light of day yet as drugs, so I'm certainly not going to tell you to prescribe anything in particular. And the focus of my talk is going to be on therapeutic insights we've obtained from trying to study mice lacking individual integrin subunits. So to give you an idea what I'm going to talk about, I first have to introduce the integrin family, which is the family that my lab's been studying for nearly 30 years now. And integrins are alpha-beta heterodimers, so each subunit spans the membrane once. There's a large extracellular domain for each subunit, and they form the two subunits together form binding sites for a number of fixed extracellular ligands. And on the cytoplasmic face, integrin subunits have an amazing capacity to assemble multi-protein signaling complexes. The simplest version of those complexes is shown here, and this is a function of integrins that's conserved across all multicellular organisms because integrins interact with rigid components of the actin cytoskeleton and the cytoplasmic side, and rigid fixed ligands outside the cell. They're ideally situated to mediate stable cell adhesion and tissue integrity. And that's a function that's conserved from worms, flies, to humans. But you don't need very many integrins to maintain tissue integrity. In fact, in worms there are 2, in flies there are 5. But there was an enormous explosion in the complexity of the integrin family at the junction between invertebrates and vertebrates, so that in nearly all vertebrates there are 24 integrins. And certainly that's the case in all mammals. And this huge explosion in complexity was associated with integrins incorporating a number of other proteins into their complexes. So integrins now bind to several other transmembrane proteins, recruit a series of additional signaling proteins. In fact, about 90% of tyrosine phosphorylation events, a critical signaling event in cells, occurs at integrin complexes. And on the outside of the cell, integrins have adapted ability to bind to many more ligands. In flies and worms, they only bind to components of the extracellular matrix. But in mammals, integrins bind to growth factors, proteases, and cell surface counterreceptors. So they're able to take on a lot of additional new functions that are important in organisms that have multiple tissues and organs. And my lab's been focusing not so much on this conserved role of integrins in maintaining tissue integrity. Not that that's boring, but there are several other labs that do that very well. But we've been interested in what are these new functions that integrins take on? How does the new complexity that occurs in mammals, and especially in humans, play some role in important integrated physiology and in human disease? So we're interested in the cell and tissue-specific responses that integrins have adapted. And the general strategy that we've used was first, almost 30 years ago, I was fortunate to do a sabbatical in a lab of a colleague I'd recruited, Robert Patella, and our project was to, my sabbatical project was to use a new method, sounds ridiculous now, the polymerase chain reaction, to find new members of this protein family. So at the time there were 6 integrins known. We knew there had to be more. So we made degenerate primers that could amplify all of the known integrins. And during my year on sabbatical, we found 5 new integrin subunit sequences. And since that time, my lab's been mainly focused on trying to understand what the genes encoded by those sequences actually do and how they function in vivo systems. And a real breakthrough came because my lab was at the San Francisco General campus at UCSF. And at that campus, one of the other labs that was present had developed or adapted a new method for homologous recombination in embryonic stem cells. So we were rapidly able to knock out integrin subunits in mice. And then we've been studying the mice really for the last 29 years. And so the general strategy we use is to inactivate integrin subunit genes in mice. We hope that the mice live, and then we can study those mice in models of human disease. We focus on the lung because I'm a pulmonary physician, but actually the lab's been happy to follow diseases in other organs that I'm really not going to talk about very much today. So I'm showing you here in red, not black, the integrin subunits I'm going to talk about. So I'll just talk about a subset of the work that's been going on in the lab. And I'm especially going to focus on the work that's led to potential new insights into ways to treat chronic lung diseases that have been poorly treatable. But before I get to that, I just want to remind you that you've all probably used some drugs that inhibit integrin. So the first integrin targeting drug that was developed were monoclonal antibodies that block the platelet integrin alpha-2b beta-3. And that drug was used for quite some time in treating patients with acute coronary syndrome. About 5 million patients around the world have gotten this monoclonal antibody. It now has been replaced largely by better platelet inhibitors, but that was a widely used integrin targeting drug. And it demonstrated that it might be possible to target integrins to treat disease. There's also a monoclonal antibody to the alpha-4 beta-1 integrin. That integrin plays an important role in lymphocyte migration into tissues, inflamed tissues. It's incredibly effective for treatment of acute exacerbations and multiple sclerosis. Unfortunately, lymphocytes need to get into the central nervous system to kill latent JC virus infection, which about 50% of humans have in the brain. And so there have been importantly serious side effects from that medicine, but nonetheless, many patients with MS still take alpha-4 beta-1 inhibitors because they're so effective. And it's now possible to screen to see whether patients have been infected with JC virus. And the newest addition are monoclonal antibodies to a related integrin, alpha-4 beta-7. Actually, the beta-7 subunit was one of the ones that we cloned on sabbatical. And those antibodies have now been approved for treatment of inflammatory bowel diseases and are really highly effective and an important second-line therapy for IBDs. But I'm going to talk, as I said, about drugs that have not been proven to be effective yet in patients. So again, I remind you, I'm not telling you to do anything therapeutically. Really, the purpose of my talk is just to try to get people, or to share my own personal excitement with the process of basic discovery as a way to get insights into new treatments for disease. So the work I'm going to talk about started with a knockout of one of the subunits that we cloned, the beta-6 subunit, which is present in the alpha-v beta-6 integrin. And Xiaozhu Wang, a longtime colleague of mine, made this original knockout. And this was published now more than 20 years ago. And we were really surprised because with the phenotype, because this integrin is restricted to epithelial cells. We knew that from immunostaining and in situ hybridization. But the phenotype was not obviously related to epithelial cells. So the first phenotype we noticed was that these mice had dramatic inflammation in response to what would normally be minor injury. So in the skin, I'm showing here, the mice are easy to identify from their litter mates because they had these patches of baldness. This region is where the mother mice pick up their babies in their teeth. And that minor trauma led to marked infiltration of the dermis with macrophages, stained here with an antibody to a macrophage marker, F480. And they also have low-grade inflammation in the lungs that's especially prominent if the mice are kept in unventilated cages. So just the off-gassing from the bedding material, the interaction between mouse urine and bedding material causes pretty profound lung inflammation. And so we couldn't understand why knocking out an integrin on epithelial cells would cause the recruitment of inflammatory cells that don't express the integrin. But not being able to figure out the phenotype, why this phenotype occurred, we thought, well, at least we could maybe use these mice as to study in models of human disease. And we thought we would study models of lung disease that everybody knew were inevitable consequences of exaggerated inflammation. So we chose to study pulmonary fibrosis, which at that time Ron Crystal, who was the NIH, had popularized as an inevitable consequence of exaggerated lung inflammation. But what we found actually was just the opposite, which is what always makes science fun. You don't really want your hypothesis to be correct. When your hypothesis is incorrect, then you're much more likely to actually develop some new insight. And so what we found was that in wild-type mice, shown here, if we treated with a single dose of intratracheal bleomycin, the mice got modest inflammation, but quite severe fibrosis. This is just trichrome staining collagen blue. But in the knockout mice, this is the worst patch of fibrosis we could find. They basically didn't get any fibrosis at all, despite the fact that they did, as predicted, get exaggerated lung inflammation. So this combination of suppression of inflammation by the integrin and induction of fibrosis by the integrin, finally, after some years of scratching our heads, suggested how this integrin actually works in vivo. And the way it works is not by binding to components of the extracellular matrix, but by binding to a latent growth factor called transforming growth factor beta. And as I'll show you in a minute, when the integrin is bound to transforming growth factor beta and tissues are injured so that the cells, the epithelial cells that express that integrin are induced to contract, to use actin and myosin to exert force on their external environment, the latent TGF-beta has a conformational change that releases the active growth factor. So I need to give you a little bit of background on TGF-beta biochemistry to understand how this works. So there are three major mammalian forms of TGF-beta, TGF-beta 1, 2, and 3. All of them are made as large precursor proteins. And after the signal peptides cleaved off, so they're secreted, there's a large amino terminal portion of the encoded gene that is what's called the latency-associated peptide. And the reason it's called that is it gets cleaved off of the mature cytokine, shown here in green, by an endoprotease in the Golgi apparatus called furin. And then the active growth factor, the active TGF-beta, is assembled as a disulfide-linked homodimer. But the amino terminal fragment also makes a disulfide-linked homodimer that completely wraps around the active growth factor. And so before the TGF-beta gets out of a cell, it's already protected from interacting with its receptors. And the interaction between the amino terminus and the active growth factor completely prevents binding to any TGF-beta receptors. But to even make this a little bit more complicated and elegant, in every cell, there are other proteins that are disulfide linked inside the secretory apparatus of the cell to this latent complex. And these come in two flavors. There are latent TGF-beta binding proteins that when this complex gets secreted are directly cross-linked to the extracellular matrix to tether the latent TGF-beta in the matrix. And in immune cells, there are two proteins called GARP and LRRC33 that tether the secreted TGF-beta to the surface of immune cells. And that's very important because 180 degrees away from this tethering site, there's an exposed loop that contains an integrin recognition sequence, arginine, glycine, aspartic acid. So we had a lot of indirect evidence suggesting that this tethering and mechanical force were critically important for TGF-beta activation. And some years ago, Tim Springer's lab solved the crystal structure of this TGF-beta latent complex. And I'm just showing you the cartoon version of this here, which shows that most of the amino terminal fragment, shown with the two copies here in green and red, are relatively rigid. They're either alpha helices or beta strands, but the unstructured region is the region that actually makes contacts to hold in the active TGF-beta. So when force is exerted by epithelial cells contracting up here, the first thing that happens to this tethered protein is this loop that holds the TGF-beta in place opens up and releases free TGF-beta. And what that means is it's possible to regulate this important growth factor very rapidly in the extracellular space. So it takes a long time to make new TGF-beta protein and get it secreted. And cells and organisms don't have time to do that when you really need this important growth factor. And so almost instantaneously, in response to injury and contraction, you can release the active growth factor. It also means that this is exquisitely regulated in space and time. And so it makes sense because this is a growth factor that's very important both in development and also in resolution of tissue injury. So it's been long thought that TGF-beta would be a very nice target for therapeutic intervention. And in fact, pharmaceutical companies have spent billions of dollars trying to develop TGF-beta inhibitors. There are still some that are trying to work their way through the development pipeline, but I think it's highly unlikely that they're going to turn out to be used very widely. And the reason is that although TGF-beta is an attractive target because of the bad things that it does, so it contributes to fibroblast activation and collagen production, as I already alluded to, also plays an important role in tumor progression and invasion. But TGF-beta is also a critical inhibitor of early steps in epithelial carcinogenesis. And in fact, the drugs that have gotten into the clinic cause skin cancer. That's one of the reasons why a lot of them have been held. And it also plays really complicated roles in regulating adaptive and innate immunity. So in some circumstances, TGF-beta promotes adaptive and innate immunity, and other consequences it inhibits. So I was excited by the fact that we had found a way to inhibit just one fraction of TGF-beta activity by blocking its activation in the extracellular space. The other real disadvantage of trying to block TGF-beta is that there are enormous stores of latent TGF-beta in tissue. So if you try to block TGF-beta, you have to give an enormous amount of inhibitor because it's very hard to inhibit all of it. But there are only small numbers of copies of integrins on restricted numbers of cells. So it's actually much easier to completely inhibit the activation pathway than TGF-beta itself. And so that's actually been the focus of probably half of the work in my lab the last 15 or 20 years, is trying to identify ways that we could take advantage of the fact that integrins activate TGF-beta to develop more focused therapies for diseases that might be mediated by TGF-beta. And this integrin actually turns out to be an attractive target for several reasons. One is that it's expressed in normal lung epithelium, but at very low levels. And in settings of tissue fibrosis, so for example, in this patient with idiopathic pulmonary fibrosis, the epithelial cells overlying pathologic regions have enormous upregulation, 50 to 100-fold upregulation of the protein. And it turns out that this is actually a feed-forward loop, because the most potent inducer of beta-6 expression is TGF-beta itself. So a little bit of TGF-beta gets activated by the little bit of alpha V beta-6 that's present. That upregulates the receptor even more, and then there's a continuing vicious cycle that contributes to the development of pathology. And whenever we can identify these kinds of feed-forward loops, they're very attractive as targets for intervention. There's also a feed-forward loop because I told you that mechanical activation is how the TGF-beta gets activated. So as the tissue gets stiffer in the setting of fibrosis, it's easier and easier for a given signal to induce more TGF-beta activation because it's pulling against a relatively rigid substrate instead of a flexible substrate. And the other reason this is an attractive target is we immunized our beta-6 knockout mice, which is a trick we use to develop drugs. And so we could make antibodies that block in mouse and human. Actually, the antibody we used here for immunostaining was made the same way. And if we use these antibodies in a model of pulmonary fibrosis with a single dose of intratracheal bleomycin that I already told you about, and then wait 14 days and treat till there's already fibrosis established, and treat with either the beta-6 blocking antibody or control antibody, the beta-6 blocking antibody has a dramatic effect in this model. It also works quite well in models of radiation-induced pulmonary fibrosis. And that was enough to lead to a collaboration with, actually two collaborations, first with Biogen and then with a small company, Strometics, that was subsequently bought back by Biogen, to develop this humanized antibody, STX-100, that was recently found in a phase 2A study to inhibit alveolar TGF-beta signaling at an easily achievable and well-tolerated dose. We still don't know if it's effective, and that's something that I hope I'll live long enough to figure out. But one of the limitations in doing clinical trials with this kind of inhibitor or any drug to treat pulmonary fibrosis is the lack of non-invasive methods to detect early responses to therapy. So as a consequence, it costs hundreds of thousands of dollars, or hundreds of millions, sorry. Oh, off by 3 orders of magnitude there. I'm not a quantitative biologist, as you can see. Hundreds of millions of dollars to do clinical trials to test the efficacy of our candidate drugs. So it'd be really nice to have earlier readouts of whether the drug was working or not. And the other thing that's often missing in clinical trials is an early pharmacodynamic marker that tells you whether you actually effectively hit the target that you're trying to get at. And so we've been trying to take advantage of the fact that alpha V beta-6 itself is highly induced in the setting of pulmonary fibrosis to see if we could image alpha V beta-6 as a way to follow responses to therapies. And so this is work that's been done by a collaborator, Sam Gambier at Stanford. There are a couple of laboratories around the world. Actually, one of my former postdocs, Gisli Jenkins in England, has a similar probe. And this is a PET probe that's based on a modified peptide that binds with very high affinity to the alpha V beta 6 integrin. So this is a CT scan of a patient with idiopathic pulmonary fibrosis, where fibrosis is usually relatively peripheral and at the bases. And this is the alpha V beta 6 PET probe that lights up the most severely affected regions and leaves the normal apical region of the lung relatively silent. And as one really nice proof of how well this probe works, this is a patient with idiopathic pulmonary fibrosis who underwent a unilateral lung transplant. So here's the residual fibrotic lung. This is the normal, relatively normal transplanted lung. Even in a setting of transplant where there are other things going on, lung inflammation, we really don't see any significant alpha V beta 6 activity. It's restricted to the fibrotic lung. Another thing that makes this PET probe attractive is we can use it as a way to rapidly screen potential drugs. So this is just showing that if we treat mice with bleomycin and make lung slices and incubate the lung slices with the PET probe in the presence of a control solvent or a small molecule alpha V beta 6 inhibitor we've been developing, the inhibitor dramatically shuts off this PET probe, which encourages us to think that when we test this in patients, we'll be able to see whether we've given enough of the inhibitor to actually hit the target. But one limitation of the approach that I just told you about is that inhibition of this epithelial integrin alpha V beta 6 is effective in some organs, in the kidney, in the biliary tract, and the lung, but isn't effective at all in other organs that develop pulmonary or tissue fibrosis, where TGF-beta is clearly involved. And one of the best examples is hepatotoxic injury in the liver, where there's dramatic progression of fibrosis, but alpha V beta 6 isn't even upregulated in the fibrotic or fibrosing liver, and in our knockout mice or with our blocking antibody, there's absolutely no effect. So that gave us an opportunity to look for other ways by which TGF-beta could get activated by integrins to see if they would also be targets. And there were in vitro reports when we started doing these experiments that there were other integrins of the same subclass that shared the alpha V subunit that could mediate force-dependent TGF-beta activation by a different population of cells, not epithelial cells, but fibroblasts, which of course are central drivers of tissue fibrosis and are engineered to exert mechanical force on their surroundings, much more so than epithelial cells. So Neil Henderson, a really talented MD, PhD hepatologist, came to my lab on a 2-year Wellcome fellowship and decided to undertake this project. And it was convenient because he's a hepatologist, and the model we wanted to use was carbon tetrachloride-induced liver fibrosis. Excuse me. And so you can see here that we had a Cree line that could very efficiently delete alpha V from fibroblasts. This is just purified fibroblasts from the mice. It's not perfectly clean, so the line we used does target some other cells, but it's quite efficient in fibroblasts. And if we deleted the whole family of alpha V integrins from fibroblasts, we could inhibit the collagen accumulation in the carbon tetrachloride treated liver. So this is picoserious red, another way to stain collagen. And in the liver, alpha smooth muscle actin turns out to be a very good marker of the cells that make collagen, and that's also inhibited here. As parenthetically, this marker is almost useless in the lung and the kidney and several other tissues. So that's a whole other talk that I'm not going to go into, but in the liver, it turns out to work quite well. And you can see here that measuring collagen accumulation by looking at the hydroxylation of proline residues in collagen also was significantly inhibited. So that suggested that we could target some other alpha V integrin. It's not alpha V beta 6 because that's not on fibroblasts, but we didn't know what it was, but it seemed like that was viable. So that allowed us then to look back in tissues where we knew that alpha V beta 6 actually was an important target, to see whether this other integrin could also be contributing to fibrosis. So it might be more general than the liver. So we looked in bleomycin-induced pulmonary fibrosis. This is staining collagen with serious red. That's inhibited. And accumulation of collagen with hydroxyproline is also inhibited. Same thing's true in a kidney model of fibrosis, where we had already shown that alpha V beta 6 played a role. If we block this other fibroblast integrin, we could also inhibit accumulation of collagen in that model. So that's really important because it tells us that you don't have to completely inhibit TGF-beta activity in a tissue to get an effective biological or therapeutic response. That just taking the top off the amount of TGF-beta that's being activated by blocking either the epithelial integrin or the fibroblast integrin would have a dramatic, or at least a significant effect. So from these experiments, we had no idea what that other alpha V integrin was. It's not beta 6, I told you. So we were fortunate because we'd already made beta 5 and had conditional beta 8 knockout mice. We obtained beta 3 knockout mice, and we could show with a series of boring experiments that alpha V beta 5 is not important, alpha V beta 3 is not important. We did a double knockout of beta 3 and beta 5, that didn't do anything. We deleted beta 8 from fibroblasts, and that didn't do anything. So you might think that this was frustrating, but Neil's got a lot of oomph, and he was excited by the fact that we were narrowing this down. And I was excited because all I have to do is sit in my office and talk to him about it. But that left two possibilities. Either it's this other integrin, alpha V beta 1, which I'll show you in a minute. It's not genetically tractable, or there's some redundancy. And most people thought it was probably the redundancy answer, which would have been much more difficult to prove. But I gave a talk at another building at the UC campus, and I was really lucky that in the audience was Bill DeGrotto, who was a really talented pharmaceutical chemist who it turns out had 20 years of experience in pharma making small molecule integrin inhibitors. So he came up to me after my talk when I explained this conundrum that we couldn't really target beta 1 because it's in 12 different integrins. And he said, well, I could design you a small molecule inhibitor that would specifically inhibit alpha V beta 1. And I said, okay, yeah, sure, whatever, go ahead, give it a shot. And so Bill did. And together with Hyanil Joe, our talented chemist in his lab, and Neil Reed, who was a scientist in my lab, we set out to see if this was possible. And Bill already knew, it turns out, that what the chemical moieties were to target alpha V integrins, because he'd made really potent alpha V beta 3 inhibitors. And he already knew how to target beta 1 because he'd made potent alpha 2 beta 1 inhibitors that will actually come up later in my talk. So he made the smallest library I've ever heard of, which was 10 small molecules. And he told me in advance that compound 6 and compound 8 were most likely the ones to work. And that turned out to be the case. So both these two small molecules were more than 3 orders of magnitude more potent at inhibiting alpha V beta 1 than any of the other RGD binding integrins. So we used the small molecule in the same experiments. We started with liver fibrosis. We induced fibrosis with carbon tetrachloride, waited 3 weeks until they already had fibrosis and started infusing this alpha V beta 1 inhibitor by Alzit pump. And you could see that we had about the same degree of protection from liver fibrosis we saw from knocking out all alpha V integrins in fibroblasts. And we also, I'm not going to show you the data, it's all published, but you could show that the same thing happened in the lung and the kidney. So it looked like just inhibiting this one integrin pharmacologically was sufficient to explain everything we'd seen with knocking out all alpha V integrins from fibroblasts. And so that then suggested to us this model where there are two different TGF-beta activating integrins that contribute to tissue fibrosis. In many organs, this starts with activation of the alpha V beta 6 or activation of epithelial cells to contract and pull on tethered TGF-beta through the alpha V beta 6 integrin, which then presents active TGF-beta to adjacent fibroblasts and also to the epithelial cells themselves. That induces more alpha V beta 6 expression, drives this process. And then the fibroblasts also are induced to express more alpha V beta 1 than they normally do. I'm not showing you the data, but we have data that that's the case to further fuel this process of tissue fibrosis. And we don't know in advance whether people are going to resemble mice in that you could just block each one of these integrins and have an effect, or more likely, there'll be a range of possibilities in people. Some might respond more with alpha V beta or use alpha V beta 6. In others, the fibrosis might be more dependent on alpha V beta 1. So we really wanted to inhibit both of these pathways simultaneously. And it's a very long slog through the FDA to develop one drug for alpha V beta 6, another for alpha V beta 1, get each one of those approved, and then try to combine them together. But everybody thinks that combination therapy is what's going to be required to treat pulmonary fibrosis. So we thought perhaps we could use a trick and screen small molecules to look for ones that inhibit both these integrins simultaneously. And that's where this small company, Pliant Therapeutics, has come into play. And they've done a beautiful job now of screening more than 4,000 small molecules that were all engineered to try to improve pharmacokinetics and pharmacodynamics and target specific integrins. So now we have a range. This is just a heat map of a small subset of these molecules. We have inhibitors that exquisitely target alpha V beta 6. We have many that are dual inhibitors of alpha V beta 1 and alpha V beta 6, generally not targeting any other integrins. And we have some very potent inhibitors specifically of alpha V beta 1. So now we can test these single and dual inhibitors in vivo models. And just looking at the initial model I told you about, single dose intratracheal bleomycin, if we just, we have it now orally available, once a day delivery, small molecule dual inhibitor. And here I can show you that in contrast to a control inhibitor, we get substantial inhibition of bleomycin-induced fibrosis with this once-a-day delivery. Again, starting 14 days after bleomycin treatment. We can also show that this works again in the human tissue because we can take lung slices from humans with idiopathic pulmonary fibrosis and then incubate those lung slices in vitro with these inhibitors to see whether there's active ongoing collagen production that we could inhibit. And so this is just showing that in these lung slices, as we always see, there's upregulation of alpha V beta 6 on the epithelium. These slices are chock full of collagen. This is picoserous red staining of a slice. And if we incubate the slice with a control solvent, this is the amount of collagen expression we see for collagen 1A1. If we inhibit with our dual inhibitor, we get a substantial reduction, which is about the same as we can do by inhibiting TGF-beta completely with a small molecule TGF-beta inhibitor. If we just inhibit alpha V beta 6 with our blocking monoclonal antibody, for example, we don't see significant reduction in collagen. Same thing's true for collagen 3A1. So it looks like in the setting of even advanced pulmonary fibrosis that required lung transplantation, because this is an explant, there's an ongoing inhibitable process that's leading to progressive production of collagen that we can inhibit with this dual inhibitor better than with a single alpha V beta 6 inhibitor. We also used another model that in some ways maybe is more like human pulmonary fibrosis, because a single dose intratracheal bleomycin might get a little fibrosis and it resolves. But if you give repeated doses of bleomycin at 2-week intervals as shown here, the mice actually develop fibrosis that persists for as long as anybody's followed them, for 3 or 4 months at least. So in that model, which is a more severe model, we induce fibrosis, wait a week after the last dose of bleomycin, and then treat with inhibitors. And in this model, if we treat with a control antibody, that's the no effect. If we inhibit just alpha V beta 6, it's not significantly different. If we inhibit just alpha V beta 1 with an alpha V beta 1 inhibitor, it's not significantly different. But if we use the combined inhibitor, either by combining the antibody with this single alpha V beta 1 inhibitor, or by using our lead dual small molecule inhibitor, we substantially reduce pulmonary fibrosis. So this lead compound that I'm showing you here is aiming to start clinical trials targeting both idiopathic pulmonary fibrosis and another disease that's been very difficult to treat, primary biliary sclerosis, in early 2019. So I think we're on target to find out, at least in the next few years. So this one I think has a better chance of figuring out in my lifetime whether this idea actually works or not. But of course, most of these ideas don't work, right? So this is very exciting from a point of view of biology, but I'm not wedded to the idea that the drugs are going to work. We're going to have to find out by doing the clinical trials. So now I'm going to tell a completely different story. It has to do with a different human disease, asthma. And asthma has a huge healthcare burden. It's, as everybody knows, a chronic airway disease associated with inflammation and intermittent airflow obstruction. Affects about 300 million people worldwide. And it does kill some people, that's really a tragedy, but especially has a disproportionate impact on quality of life. There are many people around the world who have severe limitations from shortness of breath, and it costs billions and billions of dollars to the healthcare system to care for these patients. There are a number of drug therapies now for asthma, so it's not like pulmonary fibrosis where the drugs are just hardly effective at all. In asthma, there are a number of effective drugs. There are smooth muscle targets I'll talk about in a minute. That's what we're working on developing. The last one, muscarinic antagonists or anticholinergics, were introduced into the marketplace more than 45 years ago, before I started as a pulmonary fellow. And beta-2 adrenergic agonists, of course, are even older than that. There have been no new therapies targeting the muscle in the last 50 years. Most of the effort has been on immune targets. Of course, we've been using glucocorticoids for a long time. Actually, only 50% of patients with asthma benefit at all from glucocorticoids, but we still keep giving glucocorticoids to all of our patients. And then there's been a lot of effort to develop glucotriene antagonists, which have helped, and targeted therapies targeting Th2 immune responses. But about 1 in 10 patients with asthma worldwide suffer from what's called severe asthma. And these are patients in whom our standard therapies are not sufficiently effective to make them well. And fewer than 50% of these patients actually with moderate or severe asthma benefit from either corticosteroids or the biologics targeting type 2 inflammation. We're going to discuss one of these at Journal Club, actually, the Pulmonary Journal Club tonight. It's great to have these new therapies, but we're still leaving half of the patients with severe asthma who don't benefit from those therapies at all. And there's an increasing percentage of patients with adult onset asthma who have obesity-related asthma, which is associated with exaggerated smooth muscle contraction, but resistant to most current therapies. So the proportion of patients who respond to these new therapies is likely to decrease over time. So it's a big unmet medical need to develop better therapies. And we didn't set out to do this at all. And that's how what I do works. We're trying to figure out how basic mechanisms underlying human development, disease, normal tissue homeostasis. But if we're aware and alert, we can find clues that tell us about ways to treat new diseases or different diseases. So this is work that was done by Aparna Sundaram, a really talented person who started as a postdoc in my lab, just starting her own lab, and she's going to be the person pursuing these experiments. But what Aparna set out to do was to try to understand why the beta-6 knockout mice that we developed a number of years ago were protected in models of chronic allergen challenge. And this journey took her from this integrin to a completely different integrin that turns out to be the target that we're working on. So the basic story was that because we have things we can do to knock out mice, we usually put them through paces of several different models. And 20 years ago, we found that the beta-6 knockout mice were dramatically protected in the sort of physiology experiment that John Butler would have loved, where we're just measuring airway lung resistance in mice by forced oscillation method. We give increasing doses of acetylcholine to the mice, and normal mice, if they're not challenged with allergen, have very little response until you get to high concentrations of acetylcholine. Wild-type control mice have a big response, and the beta-6 knockout mice were dramatically protected in this model of repeated airway challenge with ovalbumin. And we had 2 center grants from the NIH over a 10-year period to figure out this mechanism, and those completely failed. But luckily, if you live long enough, you can find some creative person to come into your group and solve one of these problems. And one of the things we had done was looked at whole genome microarrays from the lung of these mice. We found lots of genes that were different, pursued many of them, found none that explained the phenomenon. But then I made one of my few actual contributions to an experiment here, which was to suggest using sandpaper on a piece of PE10 tubing to develop an airway brush for mice, like what we do in bronchoscopy in patients. And so I couldn't get anybody to do it, so I went into the lab and took a little bit of sandpaper. That was my big experiment. And a key point is putting a small platinum wire inside so that it has some degree of stiffness. We can use that brush then and just go into the airways of mice through the trachea and collect these large patches of epithelium, which is what we do, of course, in patients with brushes as well. And the reason I thought this would be useful is because alpha V beta 6 is restricted to the epithelium. So I thought perhaps there would be some gene expression signature going on in the epithelial compartment that we would lose when we looked at the whole lung gene expression. And that turned out to be the case. So from these, it was a while ago, so we did microarrays to look at genome-wide expression analysis. We found lots of genes that were different, but I'm just focusing in here on 2 clusters of genes that were especially informative. And that's this cluster here. These are genes that were induced by ovalbumin, so red means more and green means less. By ovalbumin in wild-type mice, they were expressed at much lower levels at baseline in the knockout and not induced very much by ovalbumin. And then there's another group of genes that were induced by ovalbumin in the wild type, but to a lesser degree than the basal high activity that we saw in the knockout. And so it turns out that these two sets of genes, although they were obtained from the epithelium, didn't include any epithelial genes. They were actually all mast cell proteases in these two different clusters. And of course, there are intraepithelial mast cells, but we've been totally ignoring them. We never saw this signal from whole lung microarrays. And so it turns out that there were different mast cell proteases that seemed to require alpha V beta 6 or seemed to be induced in the absence of alpha V beta 6. So we did experiments by just taking bone marrow, differentiated it into mast cells in vitro, and then doing that in the presence or absence of TGF-beta. And it turns out, as you might expect, since all this integrin does is to activate TGF-beta, that there was a huge difference whether we added TGF-beta or not. So in the presence of TGF-beta, we induced that first cluster of genes, and in the absence of TGF-beta, those genes were shut off, and we dramatically turned on expression of this other cluster of proteases. And you can see by Western blotting, this is true at the protein level as well. I've never really had results from my lab that were quite so clear-cut as this in the other experiments. And so that gave us a rationale for why this might have worked, but still didn't tell us how changes in mast cell proteases would affect airway hyperresponsiveness. But we ignored these genes because they're unique to the mouse, and we like to study human disease. And we focused on these genes because this one, mast cell protease 4, is a close ortholog of a human mast cell protease called chymase. And we went back to another set of experiments that I think John Butler would have loved, which is resuscitating a very old technique of hanging rings of trachea in a muscle bath and then studying the force that was generated by those tracheas in response, or tracheal rings in response to external agonists. And we actually used temperature-sensitive recorder to do these experiments, because that's what I had in my lab from 30 years before. We now do it with a computer, but it worked just as well with the actual recorder. And so using that approach, we could show, as expected, that if we hang these rings and incubate them with cytokines that are known to contribute to asthma. So here we used IL-13, but the same thing's true if we use IL-17 or TNF. There's nothing special about IL-13. We get a big increase in the amount of force that we can generate with these rings. And if we incubate those rings with either the mouse protease or, as shown here, recombinant human chymase, we dramatically inhibit the amount of force that we can generate from these tracheas. And the same thing's true if we use human bronchial rings. And I'm not showing you the data, but we can get rid of the epithelium from these rings, and we still see exactly the same effect. So that suggested that somehow recombinant human chymase is acting on the muscle or something around the muscle to affect force transmission. So we understood how airway smooth muscle is induced to contract. There's a classic signaling pathway, involves changes in intracellular calcium, activation of the GTPase Rho and its effector Rho kinase, a phosphorylation of myosin light chain, which allows actin-myosin bridges to form. And Aparna systematically looked through each component of this classic signaling pathway, and just like I showed you for the integrins before, none of them had any effect at all. So it didn't seem like chymase was affecting the normal contractile apparatus in the muscle. But it turns out that another lab at UCSF had done a proteomic screen together with George Coey, who was interested in mast cells, for what chymase substrates there were. And one important substrate they found was an integrin ligand, fibronectin, in the extracellular matrix protein. So that brings me back to the conserved function of integrins that I actually was mostly ignoring for the 20 years before this. And so we wanted to see whether this would actually be relevant. And so Aparna did this experiment where she incubated fibronectin with chymase in a range of concentrations, and that's shown in these dark bars. Or she incubated the airway smooth muscle cells themselves with chymase, shown here in these light-colored bars. And then she just saw whether or not the adhesion of smooth muscle cells was affected. And you can see that if we incubated the fibronectin with chymase, we inhibited adhesion of smooth muscle cells, and incubating the smooth muscle cells with chymase had no effect at all. So the chymase wasn't acting on the smooth muscle at all. It's acting on the fibronectin in the substrate. The important experiment was the next, well, sorry, there were two slides from now is the important experiment. But the first key step was to see what integrins are actually mediating adhesion of airway smooth muscle to fibronectin, and by antibody experiments, inhibiting all of the fibronectin binding integrins on airway smooth muscle cells, we could show that the beta-1 subunit's important, and so is the alpha-5 subunit. So a single integrin, alpha-5 beta-1, is the major adhesion receptor of these cells to fibronectin. So that was nice because from a therapeutic standpoint, we don't want to give people kinase as a therapy. That's not a good idea. But if we could inhibit a single integrin to affect smooth muscle forced transmission, that might be good. So Aparna used a monoclonal antibody that blocks mouse alpha-5 beta-1. It had a significant effect in this system. And if we used a small molecule inhibitor that gets into the tracheal rings better, we could have basically the same effect that I showed you before that we saw with human kinase. So that encouraged us to see whether this might actually be therapeutically tractable. And a key experiment that Aparna did was to contract mouse tracheal rings. We did the same thing with human, with high doses of methacholine to mimic severe asthma. And so under these circumstances, if we use a range of concentrations of the adrenergic agonist isoproterenol, we can inhibit only a little bit and only at high concentrations. So these are actually concentrations of isoproterenol unachievable in patients. But at every one of these concentrations of isoproterenol, simply by inhibiting the single integrin, we could enhance the degree of relaxation. So that suggests that in patients with severe asthma, adding an inhibitor of this tethering mechanism by which smooth muscle attached to the underlying matrix might actually be useful for therapeutic intervention. And then to prove that this was really related to smooth muscle alpha-5 beta-1, Aparna made a conditional knockout of the alpha-5 subunit just in smooth muscle. And in that in vivo experiment, you could see that we could significantly inhibit airway hyperresponsiveness. So we're already trying to develop with Bildegrado better drug-like small molecule inhibitors of alpha-5 beta-1 that we could either administer orally or by inhalation. And we're encouraged because alpha-5 beta-1 inhibitors have been given to patients in failed cancer clinical trials. So hundreds, or actually now thousands of patients have had alpha-5 beta-1 inhibitors without significant toxicity. But Aparna wasn't really satisfied with just stopping there because now we had a completely new way to think about inhibiting forced transmission in airway smooth muscle and treating asthma. So she went to look at what other integrins might be expressed by airway smooth muscle cells, because fibronectin is just one of the matrix proteins that the cells could be tethered on. And so there are several integrins that are expressed. This is just a PCR screen. And as a first case, she's focused on this integrin subunit alpha-2 of the alpha-2 beta-1 integrin. And so alpha-2 beta-1 is the major collagen receptor on airway smooth muscle cells. So these are similar adhesion assays showing that if we plate cells on collagen, inhibitor of beta-1 or this anti-alpha-2 antibody completely inhibit attachment to collagen. And it's known that collagen increases in the airways of patients with asthma and surrounds airway smooth muscle. So we use the same muscle bath experiment. The alpha-2 inhibitor works just about as well as the alpha-5 inhibitor, and we're just getting ready to do combined therapy. We haven't done that yet. But in vivo, the small molecule inhibitor that Bildegrado developed 20 years ago, against alpha-2 beta-1, which isn't a great drug, but it's good enough to at least prove that it works, also inhibits airway hyperresponsiveness in vivo. So this drug has an IC50 of about 10 micromolar, so it's really not the drug that we want to have, but at least it's a proof of principle that this strategy might work. So currently, we're working with Bill. We already have a library that we're developing of more selective, potent, potential oral and inhaled small molecule inhibitors of alpha-2 beta-1 and alpha-5 beta-1, which we hope sometime in the next couple of years we'll be able to at least test in patients to see whether this strategy might actually be useful. So I just want to summarize what I'm trying to get across here, which is not, again, to tell you that we've developed wonderful drugs that cure lots of people. It's really more to give you a sense of my excitement by the fact that efforts to simply understand the in vivo function of members of an important protein family, in this case integrins, has suggested new strategies that would have been completely unpredictable without just trying to figure out how things work. So nobody, I certainly wasn't smart enough to think about trying to inhibit TGF-beta activation by blocking alpha-V beta-6 or smooth muscle contraction by inhibiting these other integrins. It's only by just trying to do basic discovery that these ideas came up at all. And whether they work or not, it's exciting to think about, to see this kind of work lead to new strategies for intervening in common and poorly treated diseases. And really, whatever the outcome of the ultimate clinical trials, the possibility that one of these strategies might work has added a huge measure of extra excitement to myself and to other people in my lab to the already thrilling process of biologic discovery. So lots of people helped with this work. I got a chance to mention a number of them. Been really fortunate to have great collaborators at UCSF, so Mike Mathey, Bill DeGrotto, Lionel Joe among them. John Munger and Dan Rifkin at NYU were critical in identifying activation of TGF-beta by integrins to begin with. Sam Gambier and Richard Kitamura developed the PET probes I told you about. I've had great scientific collaborators, both at Biogen and at this new company, Pliant, and been fortunate to have a range of funding that supported these experiments. So thanks very much.